are delighted to be hosting this Ask the Researcher question and answer session as part of Bone Cancer Awareness Week. We've gathered together frequently asked questions and topics that we know are important and of interest to you, our community, and asked some of our fantastic researchers to share their knowledge and expertise. Thank you so much to everybody who has contributed to this session, and a very special thanks to Professor Gareth Veal, Dr. Lucia Cotton and Luke Farrow, our wonderful Ask the Researcher panel. As this is a pre-recorded session, we won't be able to pose any questions live, but if you do have any feedback at all or would like to ask any further questions, please do get in contact with us by emailing research@bcrt.org.uk. We hope you enjoy the session, and without any further ado, I'll hand over to our expert panel.
Hi everyone, I'm Luke. I'm a PhD student at UCL, working in the Cancer Institute. I'm working primarily on osteosarcoma.
Hi there, I'm Gareth Veal, Professor of Cancer Pharmacology at Newcastle University, and my research is involved in clinical trials of both adult and childhood cancer patients, including bone cancer patients.
Hello, my name is Lucia Cottone, and I'm an early career fellow funded by the Bone Cancer Research Trust, and I work at the UCL Cancer Institute in London on osteosarcoma research.
Before anything goes on to involve patients, we need to understand the basic biology. We kind of need to know what happens within the bone in healthy people, and then how this changes during the development of cancer. Essentially, we have to understand how the cancer survives and grows before we're able to find ways to disrupt that. And then once a viable target's identified, that's when potential treatments can be developed. And then similarly, sort of understanding the basics and the biology of the cancer and the differences between this and normal tissue can allow for easier and earlier diagnosis by knowing what to look for in patients.
To answer this question, we have to look at what happens inside the cells in our body. Inside almost every cell, there is an instruction booklet, which is called genetic material. This is unique to each person. It's a genetic fingerprint of individuals. And this booklet is exactly the same for all the cells in our body. But so how can different cells, which are so different, such as cells in the brain or in the skin, be generated starting from the same identical instruction manual? Different cells will actually use different portions, different chapters of this manual. And how they do this is because they use chemical post-its, which are called epigenetic marks, to label which are the correct chapters that they have to use. Now, cancer cells develop because they either acquire spelling mistakes in the instruction booklets, which is what we call genetic mutations, or if they acquire an alteration in which chapter of this genetic booklet they use, because these chemical post-its have been misplaced in the instruction manual. And this is what we call epigenetic alterations.
Biomarkers they can really be used to describe anything that we measure normally in a patient's bloodstream, which tells us about a disease. So in the case of cancer, they may be prognostic or diagnostic biomarkers, which would tell a clinician if a patient has cancer, and then may give an indication of how aggressive that cancer may be in the patient. The research that we do in the biomarker field is more related to looking at measuring a patient's response following a treatment with an anti-cancer drug. So they are commonly described as predictive or pharmacodynamic biomarkers. And they can tell us two things. So one is how well a patient is responding to a cancer treatment. So if a cancer drug hits the target tumor cells, then what may happen is the tumor cells may release something into the bloodstream which can be measured. So we can measure that, normally a protein, maybe tumor DNA or a fragment of the tumor cells. And we can use that to get an indication of how well the drug is targeting the tumor. And the other thing that we can look at with biomarkers is using sort of proteins that are, or fragments that are released from host cells when the anti-cancer drugs are kind of doing damage to host cells. So it may give us an early indication of which patients may be more likely to experience more excessive toxicity following their treatment with chemotherapy.
We tend to think of cancer as a single disease, but cancer is an extremely broad term and almost more of a symptom than a disease itself. It's like trying to find a cure for headaches, for example. And the way cancer develops involves an element of randomness. So even cancers that develop in the same tissue may have randomly developed different ways of growing and surviving. There isn't just one recipe for cancer. So, for example, some cancers may have randomly picked up the ability to grow really fast, but can't really move anywhere, while some may grow really slowly, but be very resilient. Like evolution in animals, really, over time, cancers pick up more and more random mutations, which just allows more and more weapons and armor to be added to their arsenal, until finally they have a combination that allows them to grow, move, and cause damage. It's these individual fingerprints or identities that we're looking for when we're trying to develop potential treatments for primary bone cancer.
Metastasis is the process whereby malignant cells from the original cancer, called primary cancer, spread to a distant site in the body. This is a complex process where malignant cells from the primary cancer need to learn to enter into the bloodstream, survive in a very hostile environment, make sure they don't get killed by the immune system, and adapt to all the new conditions of this new distant organ, which is frequently the lungs, the liver, the brain, or bone. If these metastatic cells are successful in seeding into this new soil, then they can grow and they can develop secondary cancers at these distant sites, which are called metastases. The goal, or one of the goals of systemic therapy, such as chemotherapy, is to prevent relapse and metastases. We still do not understand fully this very complex process, and scientists are actively investigating ways to prevent the formation of metastases.
One of the key aspects of cancer therapy is that there needs to be a way for the drugs to distinguish between the cancer tissue and the healthy tissue. Traditional chemotherapies tend to target some generic signatures that are common among a lot of cancers. For example, the fact that cancer grows more quickly than normal tissue, or the fact that certain mechanisms within the cell are broken. While these are often effective, they're not perfect in distinguishing their targets, and you often get collateral damage. For example, fast-growing cells in hair follicles and in the intestines that give a lot of side effects. This broadness also means that dosages have to be very carefully balanced to try and effectively take out the tumour while damaging as few healthy cells as possible. And things tend to lean towards effectively taking out the tumour while sacrificing a few healthy cells. Targeted therapies, on the other hand, are tailored to the specific cancer or subgroup of cancer, targeting a unique feature of that cancer that isn't necessarily shared with any healthy cells. These targets are usually found by studying that particular cancer type in depth in the lab and determine its unique biological fingerprint. This means that treatment is less broad and more accurate, leading to fewer side effects, more effective killing of tumours, and using lower dosages of drugs. When trying to kill a tumor, chemotherapy is like a hand grenade, whereas targeted therapy is more like a precision sniper.
So our immune system works to protect our body against infection, against diseases, and it can also protect us from developing cancer. Immunotherapy uses our immune system to recognize and attack the cancer cells. There are a number of immunotherapies developed and used alone or in combination with chemo or radiotherapy to boost the immune system to attack cancer. You might have heard of immune checkpoint inhibitors or cellular therapies such as the CAR T cell therapy. Research in immunotherapy to treat primary bone cancer is still undergoing, and these therapies have not been tested well enough. Unfortunately, what we have learned is that the immune system seems to respond to bone cancer far less compared to other cancers. And this might explain why these early trials that have been tested with standard immunotherapy have not been as successful as we would have hoped.
The majority of anti-cancer drugs that we use now are drugs that have been around for a relatively long period of time. They will target either DNA replication or cell division. And really the only reason that they target tumour cells more than host cells is because the tumour cells are rapidly dividing. So the kind of toxicities we commonly see will be things like hematological toxicity, gastrointestinal toxicity, things like hair loss. These are the kind of common cell types that divide rapidly in the body. So they're targeted at the same time as the tumour cells. So I think with the drugs that are well established and have been around for a long time, we're not going to really get away from those side effects, whereas newer drugs are going to be more targeted to the tumour and will hopefully have fewer side effects. In terms of what we can do with these well-established older drugs, I mentioned earlier about biomarkers, and that's one way we can look at, trying to use biomarkers and measure biomarkers to try to predict which patients may experience more toxicity or less toxicity, and then we can kind of make changes to treatment, make changes to dosages. And the other way to do that, I think, from my research is in pharmacology. So from a pharmacological perspective, we do a lot of research now where we actually measure drug levels in individual patients, and we can often correlate higher drug exposures with increases in toxicity in patients. So if we can measure drug levels in individual patients and personalise treatment, so we decrease the dose for patients who are getting a high exposure. On the other side, we can increase the dose for patients who've got maybe a lower exposure and maybe less likely to respond. And that's a way of, as I say, personalising the treatment and hopefully reducing some of those side effects that are commonly seen in patients.
Cancer cells continuously accumulate mistakes in the genetic or in the epigenetic code. This unfortunately means that sometimes cancer cells, or even small groups of cells within the tumor, can learn how to dodge the drugs that they have been treated with and become insensitive to them. Sometimes what changes are not necessarily the malignant cells, they're acquiring mutations, but it could also be the way in which they use nutrients in the system, or it's sometimes even in the environment that surrounds them and allows the cancer cells to become immune to the treatment. Whatever way the cancer cells find, this group of rogue cells will progressively take over and manage to grow irrespective of the treatment. And this is what we refer to as drug resistance, which is still one of the most challenging issues for clinicians and for researchers. In my early career fellowship funded by the Bone Cancer Research Trust, I'm studying what are the causes of resistance to chemotherapy in osteosarcoma, and particularly I'm looking at how the cells dynamically adapt and learn how to dodge chemotherapy by changing at the epigenetic and gene expression level, with the ultimate goal to identify a potential combination of drugs that can stop these resistant cells.
Drug development has changed massively over the past 20, 30 plus years. Certainly when I first started doing cancer research, most of the drugs in development were drugs which would target cell division or DNA replication. They would be fairly non-selective drugs, which would be useful against a whole wide range of tumor types. I think we're in a much better position now in terms of the way that drugs are developed. So drugs are developed looking at specific abnormalities of the cancer cells or the characteristics of the cancer cells, which don't have anything in common with the kind of host cells, which means you can much more carefully target the cancer cells, but hopefully not get the side effects that are seen from the more old-fashioned anti-cancer drugs. Whilst that's really good, a big problem with that is that at the end of the day, there are over 200 different types of cancer. So essentially, there are over 200 different types of disease. So, even with something like breast cancer, there are different subtypes of breast cancer. And the drugs that are now being developed will be developed specifically for a particular cancer or even a subtype of a particular cancer. And it's actually highly unlikely that that drug will then be useful for any other type of cancer. So while it's really good the way we're targeting or developing much more targeted drugs and better drugs, it means they're not going to be used as widely. And of course, that means that for those cancers that are out there where there are lots of people diagnosed with them, then drug companies are much more likely to develop drugs for example, breast cancer and lung cancer, etc.
Research in primary bone cancer is advancing, however at a slower pace compared to more common cancers. The rarity of this disease implies that it is much more difficult to obtain samples which are essential to study these tumors in the lab in sufficient numbers to test our hypothesis. And we are unfortunately still lacking experimental systems that we can manipulate in the laboratory and that faithfully reproduce what happens in the tumors.
The rarity of the cancer tends to mean that there are generally much fewer tumors around for scientists to study. And this, plus the complexity of the bone environment that they grow in, means that research into developing better treatments is pretty slow compared to the really common cancers that get a lot of attention from other charities and researchers. My PhD project in particular is aiming to find a more effective and easy way to study primary bone cancer in the lab, which will hopefully lead to easier access for researchers and lead to more precise understanding of the cancer itself.
Yeah, cancer drugs, the majority of cancer drugs that we use for many tumor types, including a lot of the bone and the different bone tumors, they are drugs that have been around for a long time. And I guess the simple answer, the reason we still use them is because they are effective. And for lots of tumor types, we have very good response rates with them. I guess the big negative with these drugs is the side effects that are seen. They're fairly non-selective drugs. So they target host cells as well as the tumor DNA cells. It's a difficult thing. I think it's easier for a cancer where response rates are not so good, that you may be more willing to look at kind of more experimental, newer drugs, etc. But I think for lots of cancer types where, fortunately, we have really good response rates, I think patients will generally be pretty happy to go with drugs which have side effects but are very highly likely to lead to a response. So I think it's the drugs that we have that have been here a long time, the drugs that are effective, they're effective for a wide range of tumor types, and they're probably going to be around for a while still, often or more often than not, I guess now, in combination with some of the newer drugs that come through.
I'm most hopeful for treatments involving the tumor environment that can often have an effect on the cancer itself. For example, recent studies have shown that primary bone cancer often recruits the help of healthy cells in order to build a little den that helps the cancer survive. The studies are ongoing at a vast pace now to assess how this communication happens and whether this can be targeted by treatments to destroy the cancer's den and leave it vulnerable to other drugs or to the body's own defenses.
the best way to treat cancer patients is to detect the cancer as early as possible. So from a kind of non-drug perspective, any kind of screening approaches that are novel, I think, are really exciting. And there is an approach called the gallery test, which has been used in the last couple of years, which is really looking at the potential for sort of picking up the presence of tumour DNA fragments in blood at a very early stage with a kind of routine test. And the NHS has been looking at this. I think this can detect kind of tumour DNA fragments from over 50 different types of cancer. I think that's something that's really exciting. So if that could be a routine kind of screening, in particularly, I guess, in older people, then that's going to make a massive difference, because if we catch the cancer early, it's obviously much easier to treat before the cancer has metastasized or spread around the body. I mean, from our own personal research perspective, our therapeutic drug monitoring program that we have, which is a national program run through Newcastle, which at the moment is focused on childhood cancer, including bone cancer patients, is really looked at this idea of personalising treatment and making sure that it's not just the getting the right drug for the right patient, but it's getting the right dose of the right drug for the right patient. So this idea of making sure we don't overexpose some patients, and similarly making sure each patient has got a good exposure to the drug, which gives them the maximum chance of responding to treatment. So this is particularly been useful over the last few years in some of the really challenging patient groups, such as infants and neonates who may be being treated in the first kind of weeks or even days of life, where it's really hard to know what dose of chemotherapy to give. But by measuring levels, we can ensure that they're going to get or target what we call the kind of therapeutic window, which is the kind of optimal exposure to the drug.
In the last 10 years, the overall cancer death rate has continued to decline. And this is largely due to the revolutionary research carried out across the world, which is improving our understanding of the causes of cancer and has transformed and expanded many treatment options. Together with chemotherapy and radiotherapy, we have seen targeted therapies, immunotherapies, epigenetic therapies being developed and showing promising effects in many different individual settings. And I believe that with the combination of multiple drugs with different mechanisms of action specific for the different cancer types, we will be able to attack the cancer from multiple angles and really see an improvement in our treatments.
A difficult question, but DNA sequencing technology and our knowledge of cancer in general is advancing at an incredibly fast rate, which means it's becoming easier and easier to determine a cancer's unique fingerprint. The timeline on personalised treatment is kind of impossible to predict, but with generous funding from charities like the BCRT allowing research into primary bone cancer, the odds have never been better.
The United Kingdom really can pride itself for the 100,000 Genome Project, a nationwide effort to decode the DNA sequence and individual mutations of rare tumors, including sarcomas. So the whole instruction booklet has been deciphered and many spelling errors have been identified. And this has translated into clinical benefits. Now, patients with primary bone cancer are offered whole genome sequence as an NHS standard of care, and the identification of specific some bone cancer mutations has allowed to improve our ability to make diagnosis. On the other hand, however, this project has allowed us to appreciate the complexity of these tumors. And we have realized that primary bone cancers are much more heterogeneous than previously thought, and that it is extremely difficult to identify mutations for which targeted drugs or already existing drugs are available. Nonetheless, I believe the more of these cancers are sequenced and the more research continues on the causes and the implications of these mutations, the closer we will get to personalized medicine being feasible.
in any area of research, international collaboration is really important, particularly for cancer, and I guess particularly for cancers which are a bit less prevalent, where we have fortunately fewer patients. Doing research either at a European level or an international level is absolutely key. So in order to have big enough trials to be able to answer the kind of questions we want to look at, which are the best drugs, which are the best drug combinations, in order to have kind of statistical power and have enough patients, we really need to do, as I say, European, potentially international studies. And the other area, I guess, is that even for kind of tumour types where we could do studies at maybe a national level, or as compared to other European countries or other countries in the States, it's still really important to have that dialogue. There's no point in, for example, us running a clinical trial in the UK to answer a question that is exactly the same or very, very similar to a trial that's been carried out in North America, for example. So that kind of dialogue, even if you're not directly working with other people on clinical trials, having that dialogue and understanding of what other groups are doing, I think is absolutely key. It's one of the things that I think we really missed with the pandemic, that kind of international collaborations. And while I think for environmental reasons, I think a lot of us have become a little bit more kind of careful about going international travel and going to meetings, it's still really important to have those kind of like meetings of kind of world experts in fields.
Especially for a rare cancer like primary bone cancer, it's critical that the relatively few researchers are in constant collaboration, not only to avoid duplication of effort, but to share ideas, results, and hypotheses, and make sure that our funding is being used as efficiently and effectively as possible.
Research simply cannot advance without sharing of knowledge and sharing of data. Collaboration is an integral part of the research process, and this is particularly true for bone cancer research, given the rarity and the high heterogeneity of these tumors. No single institution would make any significant progress independently, and it's only through international collaboration that we can share expertise, we can facilitate access to biological models and samples, and we can confirm the findings and the preliminary experiments and truly improve our understanding of the biology of these cancers.
I think identification of the role of the tumour environment in cancer development, as well as the generation of reliable and easy-to-use models of bone cancer for use in the labs. Identification of a unique fingerprint for different subtypes of primary bone cancer will allow for more directed research into personalised treatments and potentially identify new or existing drugs that can target these different fingerprints.
I would say from my perspective, of a kind of pharmacology perspective, we should be doing a lot more to try to get that balance right between the doses of drugs and the exposures of drugs these are well-established drugs. They're not going to be going anywhere in the next few years. In an ideal world, we will have new drugs which may come through in 5 years, 10 years. But I would say we need to do a lot more to not just cure patients, but cure patients with kinder treatment. So look at how we can modify dosing regimens to try to reduce some of the side effects. So I do a lot of work in childhood cancer, and fortunately, we're in a position now where I think more than 75% of children with cancers, including bone cancers, will survive their treatment across the board. But actually, around 2/3 of those patients will have toxicities that have been caused by the treatments, which are going to affect their kind of quality of life in adulthood. So I think a priority really needs to be trying to kind of maintain the good response rates we've got for lots of tumour types, but really make those treatments kinder and let the people who are surviving cancer have a better quality of life.
I believe the priorities should be improving our understanding of the causes of primary bone cancer, including the progression and the spreading of these diseases, as well as the mechanisms underlying drug resistance. The more we understand the biology of these tumors, the more we will be able to generate new ideas and offer new therapeutic approaches to ultimately improve patients' outcomes. What we have seen in the last 10 years is the beginning of an acceleration in the studies that look at the biology of primary bone cancer, and this has been possible thanks to the commitment of charities such as the Bone Cancer Research Trust and the involvement of patients who, with their generous donations of biological samples, are shaping the future of primary bone cancer research.
so much to the Bone Cancer Research Trust for all your support.
I'd like to say a massive thank you for everybody who's supported BCRT and bone cancer research. Thank you.
Thank you so much to everyone who supports the Bone Cancer Research Trust.